Our research speaker is an assistant professor from UP Los Baños. So she obtained her PhD degree in botany and MS in botany in UP Los Baños. Her research publications are extensive and includes topics like population density, floral biology, and pollination studies, to name a few. She received numerous awards, including the Best Presenter at the Virtual University Consortium Graduate Forum 2020, Outstanding Teacher Award, the 30th Founding Anniversary of IBS, Regional winner and second runner-up at the national level at CHED, at Best Higher Education Institution Research Program, and other awards since 2008. So together, let us all welcome Dr. Rodelina C. Deyto.
Hello everyone, I am Rodelina C. Deyto, a faculty from the Environmental Biology Division. I would like to thank the organizers of this event for inviting me and share my research. You have to remember that the lack of environmental education led to numerous challenges affecting the health and well-being of all living organisms on our planet. So research communication like this is a crucial step to increase awareness and to encourage our younger generations to do research. My line of research is significant to agriculture, which is important in food security and sustainability. Food security and sustainability is imperative to feed the growing population and minimize the effects of climate change. My talk for today is entitled Optimizing Fruit and Seed Set Through Floral Biology and Pollination Studies. Okay, so let us start by recalling how fruits are formed. So I have here a picture of a typical simple flower showing its different parts. Okay, so recall that fruits are formed from the ovary of a flower. But there is an important step before a fruit is formed, and that is pollination. Now, there are two types of pollination. The first one is self-pollination, wherein the pollen grains of a flower land on its own stigma. The other one is cross-pollination, in which the pollen grains come from several flowers. Successful pollination results to fruit formation. Okay, floral biology is an area of ecological research that studies the structures, behavior, and physiological aspects involved in the flowering of plants. The knowledge of floral biology is fundamental to pollination success and in obtaining higher fruit and seed set. Moreover, knowledge of floral biology is required for efficient crop breeding and hybridization programs for genetic improvement. Now, what are the areas of research within the field of floral biology? The first one is floral morphology, or the characteristics of the flower. So this is done by dissecting the flower and then examine the shape, the size, color, and its floral position relative to the plant axis. So this is the flower of tomato, and this is the flower of hot pepper. So this is the cross sections of their ovaries, and this is the stigma of tomato flower. Another one is anthesis, okay, or the time of flowering. Anthesis is the specific time or period during which a flower is fully open and functional. So how is this done? The flower is observed from bud to fruit development or until abscission. So for example, this is the anthesis of hot pepper. Okay, so at 6 in the morning, the petals were shaped like a bell, exposing the stigma and stamen. The flower was fully open at 9 and remained open until the next day with fully dehisced anthers. On the third day, the corolla and stamen of pollinated flower were removed and the ovary was enlarged, starting to develop into fruit. While flowers that were not pollinated has peduncle, calyx, and ovary that turned yellow with detached corolla, stamen, and style, and eventually fell off and disconnected from the plant. This is the figure showing anthesis in tomato flower. So the flower was fully open at 10 in the morning and remained open for 3 days. Flowers that were not pollinated eventually detached from the plant and fell off on the 6th day. A successfully pollinated flower started to develop fruit on the 3rd to 6th day after it had opened with anthers detached. Another one is pollen viability, pollen germination, and stigmatic receptivity. But first, you have to study first the structure of the pollen. So the pollen samples are mounted on slides and the pollen size, color, and structure were described. For the pollen viability is determined by staining with lactophenol aniline blue or Lugol's iodine. Viable pollen is a pollen that is competent to deliver the male gametes to the embryo sac. So on the left figure, I used lactophenol aniline blue. On the figure on the right, I used Lugol's iodine for pollen viability. Darkly stained pollen was identified as viable pollen and lightly stained grains were non-viable. The pollen grains were collected every hour from 6 to 12 noon to test their viability. And then the percent viability is computed by dividing the number of viable pollen with the total number of pollen multiplied by 100. So you just have to count the number of darkly stained pollen, then divided by the total number of pollen grains multiplied by 100. Another one is stigmatic receptivity, which is determined by getting the percent pollen germination. Stigmatic receptivity was evaluated by brushing several excised dehisced anthers on the lobes of the stigma. This was done every hour from 6 a.m. to 12 noon. And then one day after pollination, pollen germination on stigma was observed under the microscope in squash preparation stained with aniline blue. So the number of germinated pollen is counted divided by the total number of pollen grains multiplied by 100. So germinated pollen are those pollen with pollen tube. Okay, so this graph shows the pollen viability and stigmatic receptivity of hot pepper flower. The blue line shows the percent pollen viability. The orange line refers to the stigmatic receptivity. So the pollen grains were viable and fertile for 3 days, starting from the day before the flower opens. Pollen viability was highest on the 2nd day of anthesis with 96% viability with fully dehisced anthers and picked from 8 to 10 in the morning. Yeah. Receptivity of stigma synchronized with pollen viability. So the peak of receptive stigma is also at 9 a.m. But in hot pepper, their stigma is receptive even before the pollen grain is viable. So this is known as protogynous, characteristic of flower, partially protogynous. The stigmas are receptive for outside pollen for at least a short time before their own pollen is available. So this is a strategy to promote outcrossing so that other pollen, viable pollen from different flowers of the same species can land on the receptive stigma. Okay. Next one is pollination biology, which is a slightly narrower area of research within floral biology. Pollination plays an important role in flowering plant reproduction, fruit and seed formation, and for maintaining biodiversity. Pollination increased food production by almost 50%, and 30% of the global food production is a result of insect pollination, primarily by bees. So under pollination biology study, the floral visitors are monitored during the blooming period. Observations are done on the first 15 minutes of an hour from 6 a.m. to 6 p.m. And then during the observation period, the number of visits of each floral visitor on a flower is noted per day. They are identified and classified as true biotic pollinators if they visited the blossom regularly, or chance pollinators if they will not fit the mentioned behavior. So these are the floral visitors of tomato on the left side, and then on the right side, these are the floral visitors of hot pepper. Among these species, stingless bees, letter D, and Xylocopa, letter E and F, are the most numerous and the most frequent visitors throughout the flowering period. Okay, this figure shows the total frequency of visits of species foraging on hot pepper from 6 a.m. to 12 noon. A small number of visitors were observed at 6 to 7 in the morning and continued to increase thereafter, reaching a peak at 9 and then diminished until 12. The time when the floral visitors are at the highest is also the time when the pollen grains are highly viable and the stigma is most receptive. So the peak of floral visitors is at 9 a.m., which is also the peak of viable pollen, and this is also the time when the stigma is most receptive. So the foraging activity of pollinators synchronized with antithesis. The problem now is that the populations of bees and other pollinators are declining around the world due to climate change, excessive use of agricultural chemicals, biodiversity loss, and pollution. This global decline in bee populations poses a serious threat to global food security and nutrition. In order to enhance pollination of a crop and improve yield or quality, and to mitigate pollinator decline, pollination management or commercial pollination is being implemented. Honeybees are the most commonly used as commercial pollinators, but a recent report states that 46% of managed honeybee colonies were lost due to infectious diseases. There are also crops that are not pollinated by honeybees. Bumblebees are the primary pollinators used for greenhouse-grown crops such as tomatoes, eggplants, and peppers. It was observed that cross-pollination by the bumblebees resulted to 45% increased fruit set and nearly 200% increase in fruit weight. But the commercial use of bumblebee has an economic drawback. The colonies die after reproduction, so new hives need to be bought repeatedly, making this pollinator service expensive. The mass rearing of bumblebees has yet to be developed in the Philippines, hence the bees cannot be used in greenhouse pollination of pepper and tomato. An alternative species are the stingless bees, which are native to the Philippines. Stingless bees possess several biological characteristics suitable in commercial pollination. So based from my study, pollination by stingless bees increased fruit production in hot pepper by 74%. The commercial pollination with stingless bees has been practiced in the Philippines on mango. Okay, so the exact knowledge of floral biology and pollination of specific species are imperative to optimize fruit and seed set and produce greater yield. Okay, so let's help save the bees because bees matter. Okay, thank you for your kind attention.